experiment and future research, and then I'll show you my sources. So we'll start with an introduction to syphilis. Syphilis is a sexually transmitted disease that is most commonly found on the genitals. Its causative bacteria is called Treponema pallidum, and it's a spirochete bacterium, meaning that it has a corkscrew appearance, as shown in this picture. Syphilis is transmitted through friction during sexual intercourse. So Treponema pallidum enters through the worn down area on the genitals after friction has occurred in sexual intercourse. Also, it can be transmitted vertically from mother to child. There are 3 different stages of syphilis: primary, secondary, and late. We'll start with primary syphilis. So this is the first stage of syphilis, and atypical cankers, as shown in this picture, can appear after 10 to 90 days after infection. These cankers subside 1 to 4 months after they have shown up, and sometimes lymphadenopathy can appear, which is the swelling of the lymph nodes. Secondary syphilis is the second stage of syphilis, and symptoms include orogenital mucosal lesions, such as these, rash, fever, headache, and organ issues like glomerulonephritis, and hepatitis and arthritis. So this is an example of rash. And glomerulonephritis is enlargement of organs. So late syphilis has 4 types: gummatous, cardiovascular, neurosyphilis, and meningovascular. So gummatous syphilis can have enlargement of organs, ulcers, perforation of or destruction of the nasal septum, which is muscle in your nose that separates your 2 left and right nostril airways. And this is an example of a destroyed nasal septum. And cardiovascular syphilis has symptoms such as destruction of the elastic tissue of the aorta, which is aortitis, which is inflammation of the aorta, and aneurysms, which is unresponsive nerves in the brain. Another part of late syphilis is neurosyphilis. This is neurosyphilis, which is damage in the brain. Symptoms of neurosyphilis can be blindness, dementia, irritability, headache, confusion, poor concentration, depression, and 6 other symptoms that are much more complex to explain. Meningovascular syphilis can include cranial palsies, which can be when your brain can cause paralysis, and stroke syndromes, which is when a blood vessel in the brain pops and there's a lack of supply in the brain. So now we'll go into diagnostic methods. There are 3 different methods of diagnosis: dark field microscopy, non-treponemal tests, and treponemal specific tests. Dark field microscopy is the most accurate when an active canker is apparent. So first the canker is cleaned and then swabbed, and that sample is put on a plate and put beneath a microscope with a dark field condenser. Pictures such as these can appear. As you can see, there are spirochete bacterium in the picture. And before the exam can be deemed as negative, there must be 3 negative exams on 3 different days. Non-treponemal tests are based off of detecting biomarkers that are released during cellular damage that occur from the syphilis spirochete. Examples of non-treponemal tests are venereal disease research laboratory tests, rapid plasma reagent tests. The accuracy of this test is questionable because it depends upon who is conducting the test. Treponemal specific tests detect antibodies that respond to the infection. So just like in dark field microscopy, the canker is cleaned, swabbed, and then put on a petri dish along with antibodies that will respond to syphilis. And if these antibodies do respond to syphilis, then it is positive, and if it does not, then it's negative, most likely. Types of these tests are enzyme immunoassay tests, T. pallidum hemagglutination tests, microhemagglutination tests with T. pallidum antigen, fluorescent treponemal antibody absorption tests, and enzyme-linked immunoabsorbent assay tests. The recommended tests for each stage of syphilis: for primary, dark field microscopy is suggested. Secondary, non-treponemal tests and treponemal-specific tests are needed to confirm the non-treponemal. And for late, because the symptoms are mostly internal, chest radiographs and X-rays are needed. So treatments. The most common treatment for syphilis is penicillin, and it has been known to cure early stages of syphilis. If syphilis is left untreated, it can advance through the stages, so it is needed to treat it early in order to keep from getting to late syphilis. For primary syphilis, penicillin G benzathine is needed, and if a patient is allergic to penicillin and is non-pregnant, treatments such as doxycycline, tetracycline, ceftriaxone, and azithromycin is needed. And for secondary syphilis, the same treatments are used as primary syphilis. For late syphilis, penicillin is once again used, and for those who are allergic to penicillin and are non-pregnant, tetracycline and doxycycline are used. For neurosyphilis, because it is more severe among all of those of late syphilis, aqueous crystalline penicillin is used, and other versions of penicillin, such as penicillin G propane, are used through IV. So now I'll discuss a historical experiment that was done after the Great Depression called the Tuskegee experiment. So in 1905, T. pallidum, the causative bacterium, was discovered, and until around the Great Depression, it was just something to be afraid of. It wasn't a big epidemic that occurred. From 1929 to 1941, the beginning of the Great Depression occurred. So syphilis became more prevalent during the Great Depression because of low income. So for families who had low income, they were unable to get treated, and it was killing people. So in 1932, the U.S. Public Health Service noticed this and partnered with the Tuskegee Institute to study untreated syphilis. The purpose of this experiment was to determine the prevalence of syphilis among rural blacks in the South and to explore the prospects for mass treatment. The methodology was they took 399 impoverished black men with late syphilis in Macon County, North Carolina, and they used 201 controls who had never had syphilis in the first place. These subjects were tricked into believing that they were receiving health care and were not having their blood taken from them for testing. When penicillin arose, the subjects were prevented from getting it at all, and as a result, 3/4 of the 399 black men died, while others infected their families. The response was in 1972, the project was exposed by an ad hoc committee and considered both a failure and a disgrace to the medical community. A $1.8 million class action suit was filed against the government for the disgrace that they had created. For current research that is occurring, many people are conducting reflections on the Tuskegee experiment. They're referring to the ethics and the information that was gained, though there is not much. Syphilis, and they're also studying syphilis in connection with HIV. They're noticing that those who have HIV are showing symptoms of syphilis in addition to HIV. These are my acknowledgments, and these are my sources. Thank you very much. Question, comments, constructive criticism? Experiments being conducted, and it's dissolved after the issue is finished.
What does meningovascular mean?
Meningovascular is just a type of syphilis. I didn't really find out what it meant, besides the fact that it's a type of syphilis.
Yes.
In your research, did you come across any sort of like public health measures that are being taken or initiatives that are trying to stop the spread of syphilis through like education or something like that?
They're educating people in schools, so in schools they're having better health classes, informing their kids about like what syphilis is and that it's a risk when doing unprotected sex. But otherwise, there's not a lot that's going on that I came across in my research.
Any other questions? No?